Good morning, everyone. My name is Stephanie Hogan, and today I am doing a demo video about bacteria. And I think I'm just going to pretend that you are my class. So let's get started. At the beginning of each lesson, I always have a warm-up. I call it a catalyst, but it's also called a warm-up. And I have a total of 5 catalysts in a cycle. And so the catalyst today is one of 5. Today the students you will have to answer this question, and I always have these questions typed up on a PowerPoint clear for all the class to see. So today the question is, decide if the statements are true. If they are not true, please correct them. Number 1, if an object cannot grow, it is alive. Is this true or false, and why do you think so? And I would wait for a student response. So maybe someone would say true. And if a student said true, I would say, why do you think it's true? Or if a student said it was false, I would say, why do you think it's false? And then sometimes I would generate a classroom discussion based on if there was some confusion on the question. Number 2, viruses are alive because they can reproduce. True or false? I would wait to see if any of the students would answer. Call on the student if no one raised their hand, and ask if they think it's true or false and why. Number 2 is false, because viruses cannot reproduce by themselves. They only reproduce if they have a host cell. Number 3, living things are divided into 3 kingdoms. True or false? Students would answer, or whatever they would answer. The answer is false, because there's actually 6 kingdoms that all living things are divided into. After the catalyst, which usually takes about 5 minutes, I would then have the students look at the vocabulary on page 16 of their textbook. There are 5 vocabulary words: bacteria, archaea, producer, decomposer, and parasite. I would put the students into 5 groups. Each group would have their own word that they would have to look up the definition using their textbook. When the group has found their definition, I would have them write the definition on the board, and then also they have to use the word in a sentence. Just so that they're applying that word outside of the definition. And I said that would take maybe 7 minutes or so. After that, we would go through the definitions together. I'd have the students write them down in their class notes. After that, I would do a mini lecture, no longer than 5 or 10 minutes talking at one time, because I think if teachers talk too long, they will lose the interest of the class, and that's not what I want to do. I want to keep the students engaged and keep them interested in what I'm talking about. So I would begin talking about bacteria, and that bacteria are everywhere. You can find them everywhere. And then I would ask the students, where do you think you could find the most bacteria? Or where do you think you could find the least bacteria? Where do you think most bacteria are found? And maybe kind of have a little bit class discussion about that. Keep going with that concept, though, that bacteria are found everywhere on Earth, and that they are the simplest living thing on Earth. That's the most important, that bacteria are the simplest living thing, that they're found everywhere. And bacteria are so small because they're made up of only one cell, and they don't have a nucleus. So that's why they're so small. And then I would say that they reproduce using binary fission. Bacteria cells can be 1/10 to 1/20th the size of other cells, of animal cells, plant cells, so they're really, really small. Bacteria have cell walls, which make them more complex than viruses, and that bacteria come in different shapes. They can be spiral shaped, rod shaped, or they can even be round. I would then go in and talk about archaea, and that archaea are similar in size to bacteria, but they have more characteristics and are more common to cells in complex organisms. They can survive in extreme environments, and they're often grouped according to where they live. That's the most important thing, because there's different types of archaea. There's methanogens, halophiles, and thermophiles. And based on the type of environment that archaea live in, that's what group they're sorted into. Excuse me. After I was done talking about those two things, I would have a quick questionnaire going back to the objectives written on the board. One of the objectives is why? Why are bacteria so small? How can they be so small? And then I would have the students write the answer in their notes, right underneath what we've been talking about. And I would also want them to answer, what are the two types of single-celled organisms? Because we have just discussed those two things, so it's good for them to get that in their mind, reoccurring, so they're seeing it and they can recognize that they've just learned those two objectives. I would have them answer the questions individually first, and then have them discuss it with a partner. After that, I would have them report out the answer, and see if everyone is grasping the concept or if they're not grasping the concept. I would then move on to the third objective of the class today, which is how bacteria help and harm other organisms. Bacteria are helpful because they break down matter in dead bodies and waste material. Cities use bacteria to break down sewage and make the water clean again. Help make nitrogen available to plants in a form that is useful to them. So bacteria help and harm other organisms. And they're harmful because they can invade our body, multiplying in body tissues and dissolving cells. They make us sick. They can poison our body with chemicals they produce and release. They can poison our body with chemicals that are part of the bacteria themselves. For example, Salmonella is an example of this. So at that time, I would tell them that they can either close their notes because we're going to move on to something new. Something I like to do if there's ever like a little question activity in the slideshow that I was provided, there were some questions. So I decided to make up a little scavenger hunt activity. I made there's 13 questions, so I made 13 little note cards. This is question number 1. It says, how does a bacterial cell compare in size with other cells? And number 2 is, why are bacteria thought to be so simple? I made 13 of these, and I would tape them all over the classroom. And I would have the students just simply go and answer every single question, and all the answers can be found in their textbook. When they're all done, or if some students get done before, they can start working on the homework, which is also written on the board. Or if they take the whole time to work, that's fine too. You can also make it fun by saying the first 5 students done get a prize. And the prize can be an extra credit point on assignment, a piece of candy, a new pencil, something like that. So when all the students are done, the questions and answers will be discussed. After that, I would do the wrap-up activity, which is the third objective question, and the students have to answer that before they leave, which is, how can bacteria help or harm other organisms? And then I would just make sure all the students are aware of the homework posted on the board. Thank you so much for watching. I appreciate you watching my video. Thank you.